Hello friends, welcome to my YouTube channel called Everyday Manufacturing. So today I have brought a lecture on a topic called ISO 11737-12018. This standard is mainly for the sterilization of an healthcare product based on the microbiological method. Part 1 speaks about the determination of a population of a microorganism on the products. So my friends, इस standard में there are mainly 9 clauses, right? Each clause deals with certain of the aspects. So let's go clause wise in the standard in order to understand the standard easily. Clause number 1 speaks about the scope. So this standard mainly specifies the requirement and provide guidance on the enumeration and microbiological characteristic of a population of a viable microorganism present on a healthcare product or the component or the raw material or the packages, right? So ये जो standard है इसका जो scope है ना, this scope mainly speaks about the number of microorganisms, I mean the viable microorganisms which are present on the any particular medical devices, right? So either the devices, either the component, either the raw material or the final packaged good. I hope this is clear what clause number 1 scope speaks about. Clause number 2 is the normative references. So this standard is in subsequent reference of a certain other standard. The first one is the ISO 10012, which is Measurement Management System. ISO 13485, which is a medical device quality management systems requirement for the regulatory purpose. ISO 15189, which is medical laboratories. ISO 17025, which is general requirement of the component or the testing and calibration laboratory. So mainly this standard is in reference lieu with the 4 of the other standards. Clause number 3 speaks about the terms and the definitions, right? So the terms and definitions such as, for example, such as what is batch, right? What is bioburden? So my friends, batch के बारे में तो आपको सबको पता है. A batch means a X amount of quantity of a particular product manufactured in single shot. I think manufactured. That is called a batch, right? Bioburden. So my friends, to understand this standard, you need to be well versed with the bioburden. I mean, what is bioburden? How to conduct the bioburden test, right? On the basis of which factor this bioburden depends upon, right? So you need to understand this in detail, which I am going to speak in today's lecture. So bioburden. Bioburden means a population of a viable microorganism which are there on a product or in a product, right? It is clear. Bioburden correction factor. So my friends, once a medical device get sterilized, then after sterilization, how much amount of microorganisms are still there, right? So that amount is represented with a correction factor. I mean the bioburden correction factor. A factor by basis of which you need to re-correct your bioburden. I mean you need to reduce the bioburden. You need to go with the bioburden test once again, right? Clear. Fourth one is the bioburden estimate. A value representing against the bioburden correction factor is called as a bioburden estimate. I mean the amount of viable microorganism present on that product. Fifth one is a bioburden method suitability. Sixth is the bioburden spike. So let's say you have done a bioburden test, there are series of data available. So a particular data which Spikes the bioburden test. I mean, let's say there are 5-6 types of microorganisms which are there on that medical devices which are viable in nature. So among those 6 organisms, there could be a probability that one of the organisms will show a spike in the bioburden value, I mean the bioburden test. That is called as a bioburden spikes. The 7th one is the correction. You must be knowing the what do we mean by correction, corrective action. So I am not going to speak this definition in detail. The 9th one is the preventive action or the culture conditions. The 10th is establish, and so on. So my friends, please go to this standard and look up on the terms and definition. If you are not able to understand with the terms and definition, please do reach me and comment in my description with the below link. So clause number 4 speaks about the general requirements. So clause number 4 में there are sub clauses. Clause number 4.1 speaks about the documentation. So my friends, whenever you do your selection for medical devices, then you need to document the processes, right? So in this, you need to have your certain procedure or the SOP in which a bioburden will be defined. I mean, how to detect the bioburden test and all, right? The clause number 4.1.2 states that you need to record each and every processes, and this record need to be approved by the designated personnel, right? Similarly, in reference of the ISO 13485. Then clause number 4.1.3 and 4.1.4 speaks about how to do the records in terms of the observation, in terms of the calculation, in terms of the data derived, and then how to transfer this calculated data to for an appropriate checks. This was the clause number 4. Clause number 4.2 speaks about the management responsibility. Again over here, in similar to the ISO 13485 standard, management is also equally responsible to implement the proper process related to the sterilization of the medical device. So MR plays an equivalent role in this standard. Clause number 4.3 speaks about the product realization. So my friends, keep in your mind in ISO 13485 what we were exactly meaning by the product realization. Similarly, it applies over here in this standard, right? Then clause number 5th speaks about the speaks about the selection of products. So my friends, I'm not going to talk about clause number 5, 6, 7, 8 and 9 in today's lecture, but before taking this lecture in subsequent slide or in subsequent lecture, let's discuss about the bioburden test. What is this bioburden first of all? So I'm going to explain the meaning of bioburden with a simple footage which I'm going to display on my back side of my screen. Please look at that process carefully and I'm going to explain you on each and every step. So let's start that footage. So be very confident in that. Before starting the bioburden testing of your biosafety cabinet, operator must have a proper protective clothing in form of lab coat and sterile gloves. Open the designated position of the biosafety cabinet, followed by turn on the blower and the UV light. Now wipe your biosafety cabinet with a 70% ethanol in an unidirectional, as you can see in the footage. Now the time comes where you need to wipe even your culture dishes too, and it should be wiped again with the 70% ethanol, followed by placing in the biosafety cabinet. You need to ensure that your culture dishes should be placed evenly in the biosafety cabinet. there should be 4 dishes for the 4-foot cabinet or 6 dishes for the 6-foot cabinet. Mark the biosafety cabinet number followed by date, site as well as the timing on the Petri plates. Open the cap and avoid hand crossing over while opening the dishes. Each culture dish were opened and were kept for a certain amount of time. After exposure, cover the cap of the culture dishes once again. Record the finish time and seal the culture dish with the parafilm. All these culture dishes is now to be incubated in upside condition at a 25 degrees Celsius for at least 3 days. Observe and record for any bacterial colony on the agar plates. Move the dishes to 30 to 35 degrees Celsius incubator and culture for at least 2 more days. Observe and record for any bacterial colony on agar plates. There should be no bacterial colony should be observed in order to meet the eligibility criteria. So my friends, I hope आपने मेरा footage देख लिया. Now you are able to understand what does this footage exactly means upon. I hope you guys have much enjoyed my today's lecture. Please do subscribe my channel, like, share and comment against my video. Take care. Bye bye.